Imagine a grey oak tree. All appears healthy and ordinary from the outside, but all may not be as it seems. Sometimes things appear orderly from a distance, but it's not until you focus your attention to the small details that you find the truth. The same is true for science. Alzheimer's disease is a prime example. Alzheimer's disease is the most common form of dementia, affecting approximately 25 million people worldwide. Alzheimer's disease is characterised by the formation of plaques and tangles of proteins within the brain, resulting in damage and loss of brain cells, which drive drastic cognitive impairments, which can significantly impair an individual's quality of life. Prior to the onset of typical Alzheimer's disease symptoms, such as memory loss and language problems, a patient may appear fine from the outside, but upon closer inspection, the brain is actually undergoing drastic changes. Plaques made from the protein amyloid are slowly forming. These plaques alert the brain's first line of defense, the microglia cells. Microglia act to protect the brain from harmful objects, to keep the body's control center functioning. Microglia migrate to and surround these plaques, and on the surface appear to reduce plaque formation by engulfing plaques and releasing plaque-degrading proteins. However, evidence suggests there is more to this story. Microglia are believed to change during Alzheimer's disease and become harmful by releasing damaging compounds such as interleukin-1 and reactive oxygen species, resulting in the loss of brain cells, which then worsens the symptoms of Alzheimer's disease. To understand how these microglia change and how these changes could influence Alzheimer's disease, we must focus our attention to the minuscule changes occurring within the brain. This is where exciting new technologies come into play. Brains of Alzheimer's disease mice can be cut into thin slices using sharp razors, which can then be stained with agents such as Congo red, which selectively stain plaques within the brain. Scientists can then hunt down these red plaques and use lasers to dissect and isolate the plaques and their surrounding microglial cells with a technique known as laser microdissection. Using these plaques, we can run a procedure known as qPCR to see how the expression of thousands of genes change in microglia in response to Alzheimer's disease, giving us an insight into the real story behind microglial's impact in Alzheimer's disease. My name is Sam Drinkle and I'm a master's student at the University of Manchester.